I think we can just go straight to Giulio, who will be talking about generative modeling of adaptive immune receptor repertoires with the tool SONNia, and he's currently a senior machine research scientist at Imprint, and before he was a Humboldt Research Fellow at the University of Leipzig, a postdoc scientist at UC Berkeley, and a PhD student jointly at ENS Paris and MPI for Dynamics and Self-Organization in Göttingen.
Okay.
Floor is yours.
Thanks. Let me share. We have the keynote. We have here. You're able to see my cursor?
Yes.
There's the pointer. Okay, thank you very much for the introduction. So, yeah, I'm Giulio, and today I'm gonna talk about some work that I've been doing during my PhD under the supervision of Alexander Walczak, Thierry Morat, and Ramita Norman. And the title is Generative Modeling for AIRR-C with SONIA. So we know that our adaptive immune system is really good at recognizing novel invaders, and is able to do that thanks to the power of randomness. So we know that there is each naive T and B cells expresses its own receptors, and the repertoire of these receptors is generated through a process called VDJ recombination. On top of that, selection in T cells in the thymus, and B cells because of central tolerance reduces diversity and protects us from autoimmunity. And so we have, if we try to characterize these two processes using a modeling-based approach, we're able to put some numbers to the whole thing. And so we know that the potential diversity coming from VDJ recombination and selection is around 10 to the 16, 10 to the 18 cell unique receptors. What we observe in a single individual is a subset are only around 10 to the 9, 10 to the 11. And what actually we are able to extract from a single bulk sequencing experiments is between 10 to the 4 to the 10 to the 6 unique receptors. And so we have this multiple level of sparsities where in order to bridge this gap, a model-based approach could really help. And so the privacy of the repertoire is something pretty much well understood. We know that because of this first level of sparsity, like from the 10 to the 18 to 10 to the 9, 11 that are expressed in an individual, there is a very low probability that two randomly sampled receptors are found in two random individuals. And here you see the distribution for T cells and for B cells of the log probability of after selection, post-selection probability. And let me see. And then we see that for T cells, the probability in general is slightly higher than B cells. So B cells are more diverse, and so the degree of overlap is even lower. And not only that happens, but also we know that if we look at a cohort of individuals and we infer models of VDJ recombination and selection, we see that there is variability in the generation itself. So the mechanism itself of VDJ recombination varies across individuals. And many people in this community have been also working on trying to find a relationship from that to the genetic background of an individual. And so back to VDJ recombination, we all know how it works. We have some V genes, D genes, and J genes. We choose one random one from them. We do some deletion of random length. We add some, we insert some nucleotide inside, and we get this nice, beautiful sequence. And the lab that I entered has been working for many years in trying to characterize this using a physical model called Igor, where Igor, what really does is really following all this process. And for a specific receptor sequence, tries to look at enumerates all the possible processes with a longer deletion, shorter deletion, a little bit more insertion, a little bit less insertion, and sums up all the possible processes that are consistent with whatever the sequence that we're able to see. And this is what is called, what allows us to give like this generation probability. On top of that, there is selection. In case, as we were saying, in case of the T cells in the thymus, Here we have, through the years, first proposed by Elanat in 2014, the first selection model, and then I entered the lab really starting to work on the selection model and try to expand them. What we do is to really look at the amino acids receptor sequence of the TCR or the BCR, look at the V and the J genes and at the CDR3 length, and try to understand what is the difference between what is the underlying expectation from VDJ recombination and what we actually observe in a specific subset, T cell or B cell subset, or in the peripheral blood. And what really these selection models do is that they have some expectation here in blue from PGen. The data looks as that, so the expectation is from the CDR3 length distribution, and so peaked around 15. But we see that from the data, the CDR3 distribution is slightly different, and what the selection model does is learning a weight that pushes us from the generation to the selection. And what SONIA really is just a linear combination of features. It's a linear model, a linear set of weights that for each one of them pushes up the distribution up and down. And so it's as we were seeing also from the talk before, it's hard somehow to look at unsupervised models and try to absolutely test whether they're working or not. But one way to do it is to look at the public sequences, as they were looking at as well. And so here we're looking quantitatively really at Emerson data. So it's data from 700 individuals. And we're trying to compare receptors that have high probability from the model in these on the y-axis versus how many times are seen in the data. And the two correlate very well, and this is really in log scale. So this is really, really nice to see. And what we can do is to compare to other more neural network-based approaches to do the same kind of test. So for example, using variational autoencoder from that use this autoencoder architecture to then sample receptor, compute probability. So in principle, they give they're able to be very flexible and be able to get the whole distribution. And when we look at the performance in characterizing this public receptor, we see that here is we compare the performance using Kullback-Leibler divergence, that it's better if it's smaller. And so we see that these linear selection models are really working pretty well and better than powerful neural networks. And so, and this somehow suggested us that we already have a good starting point, but the best case scenario is whether we can find a way to combine these two worlds. On one side, we have this Igor model, this biophysical model of VDJ recombination that we really trust, that we are characterizing VDJ recombination very well. On the other side, timing selection, central tolerance are very dynamic and nonlinear processes. And so a simple linear model might be not enough to characterize them. And so what we want to do is, can we expand from a linear model to a nonlinear model? And so that was really what I've been working for the major part of my PhD, is to developing this deep selection model and testing them. And so the idea is that before we have our input to the model, that is CDR3 length, VJGs, amino acid, and we have one weight for each, as in a linear model, to get some energies. In the neural network case, the input is the same, but there are multiple layers that try to capture also covariation across this input receptor sequencing. And long story short, here again, Kullback-Leibler divergence to measure performance, we are able to get much better into characterizing the repertoire. So another thing that these neural network model work really well on is to look at interchain correlation, because linear models by constructions, they can't get correlation between input feature. But when we look at pair chain data, where we have both, for example, alpha and beta chain in the case of T cells, we wanted to try to understand whether there is some covariation across the chain and whether we are able to describe it. And so what we did in order to get there is to build first a model for the generation of these receptor sequences. And so we have a PGen model for the alpha chain, we have a PGen model for the beta chain. And this is independent generation. And so we have just the product of the two. And then what we want to do using our nonlinear models is to get a nonlinear selection that gets as input the information of both the alpha and the beta chain and tries to get covariation. So before I was showing you T cells. Now I go back into the case of P cells. And so here is a antibody heavy lambda data, pair data. And there is a lot going on in this plot. What I want to show you is that, so we are looking at mutual information between some features. And there are different kind of features, such as the J gene and the CD3 length of the lambda chain, or the V gene and the CD3 length of the heavy chain. So here we have all the intrachain features, and the data we have in blue, which is and we see that many different versions of the model that is linear or non-linear, they are all working very well. But for getting interchain correlations right, only these deep joint selection models are able to capture that. And so this was so far so good. But the question is, what can we do more? Can we learn something about the biology here? On one side, we have these generative models that work really well on receptor repertoires, but also what it's useful about this is the fact that there are also very data efficient. So we can look at a specific sample and build up a very specific model only for that subset and repertoire. And so this is what we have been looking at afterwards in collaboration with the Klatzmann lab. And so there we had access from the thymus of 9 individuals, 4 different subsets. So the double positive stage of T cell development, CD8 positive, regulatory T cells, and conventional T cells. And so since we have these 9 individuals, we could learn a different PGen model for each of the individuals, so personalized generation model, and then a different specific selection model for each of the T cell subset. And so this gives us a big list of models. And then what we can do is computing distances. So here it's what is called the Jensen-Shandon divergence between these two models. And what is good about this is that unlike Jaccard index or any of those distances that are based on clonal overlap, this kind of distance doesn't need any clonal overlap between repertoires. It's just like looking at the receptor sequences, scoring them using PGen, and looking at the difference in scoring between two models. And this is how you get a distance out of it. And so you can apply it also to smaller data sets. And so by doing that, we have many models. So the big distance matrix. And the first thing that we are doing is mapping them down to low dimension, because there is a little bit of structure, but we want to visualize it. Turns out that the most reasonable number of dimensions that allows to describe this matrix were 3. And so here I'm showing the 3 dimensions. So here in the first 2 dimensions, what you see is that each of these single point is a repertoire. And so we have the PGen repertoires for the 9 individuals, and they are connected by a line if they are belonging to the same individual. So we go from this PGen repertoire to the double positive stage, then we have this CD8 positive stage, and the 2 CD4 positive stage clustered a little bit together, so it's harder to separate the 2. On the other side, on the 3rd dimension, what we see is that it very much aligns on individual variability. So there is really these initial variabilities that is defined by PGen is able to see, we're able to extract it from this big distance matrix. And so these are visualization. We can also start quantifying a little bit more what's going on here. And so here I'm computing the diversity using the entropy, this is really the entropy of the model. So that measures how many unique sequences we could generate from this model. And so we know that starting at the level of VDG recombination, we have the highest diversity. As we go through selection, this diversity is reduced, and turns out we get some around 31 bits of entropy at the level of the single positive stage. And this didn't have to be, like diversity could have increased, like you have some distribution, could have stayed the same. The fact that it is reduced is because of an effect of rich getting richer. So it has been already shown before. Now we see it, we really stratify it at the level of different T cell subsets is that there is this mechanism where if you have a higher chance of being generated, you also have a higher chance of passing selection. And so this is already established at the level of double positive stage, where there is this positive correlation between generation probability and selection, and we see it even stronger at the level of single positive stage. And that is really like what brings this reduction of diversity down, where those sequences that have high probability, they get even more stronger. And the tail of this distribution, this low probability sequence, are getting lost. Another thing that we can do is that we can look at within this big matrix of distances, we can look at different cell types coming from the same individual, or the same cell type coming from different individual, and try to characterize all these different distances and try to put them all on the same scale in this case. And so, for example, here, for the case of same cell type, different individual, here we see that across individual, there is some diversity that is established at the level of generation. So individuals are different, and so there is some distance of 0.1 across them. And as T cell maturation happens, this doesn't change much. So it really means that this individualization of repertoires is established at the level of a VDJ recombination, and selection just let this repertoire stays more or less independently different. But then on the same axis, we can check about within one individual different cell subset. And again, here there is a lot of information going on. Like one interesting observation, for example, is that conventional regulatory T cells, they are very, very similar within one person, and they end up being much more similar than two different regulatory T cells subset across individuals. So it's nice to have the possibility of comparing all of that. Another thing that you can do with these models is that once you have a p-post model or like a SONIA model for each of the subset, you can think about a mixture. Like you think about in the case where you don't have the subset anymore, where you have a mixture of CD4 and CD8, and you can infer the fraction of the CD4 and the CD8 in this mixture. So the idea is that you have a model for CD4, a model for CD8, and then in the data you're looking to maximize this frequency f, that is the fraction, from maximizing this likelihood. And so we tested this by creating different synthetic mixtures of CD4 and CD8, and we show that we can recover very well the fraction. Another thing that you can do is to look at the local neighborhood. So far we have been shown like this VDJ recombination model, these selection models that are trained on these broad summary statistics of the repertoire. And so it's not obvious that by looking at this global statistics, once you go focusing on a single T cell receptor or B cell receptor, and you look at the local sequence space, so in this case, all the 1-point mutations, it's not obvious that we should be able to characterize it very well. And so what we can do is just computing probabilities again. And so here we are looking at our receptor sequence, and we're looking at whole point mutations, and then we compute the average probability of that. And that is, by doing that, we can compute the predicted number of just neighbors that this receptor should have. And then we can look back into the data and try to see whether this predicted number of neighbors follows the data. And in this case here, we're looking at the prediction of our deep neural network model, and we see that the correlation between the prediction on the number of neighbors from the data and the model is very, very good. And it's actually up to whatever is the upper bound that comes from estimating the noise in the data itself. So it's as good as it gets in practice. And this is much better than having linear model. That is much better than having generation models. So it's really in this try to get this nonlinear covariation of variables in the receptor sequence that we are able really to extract a lot of information about the the neighborhood of a receptor sequence. And there are many other interesting tests that I've been doing. So I just like the paper came up this year, and so you can have a look at it. But an obvious application of all of this is that it has been shown already before using generation probabilities that if you look at the repertoire and you look at some cluster of receptor sequences, there are some that are just by pure chance, and there are ones that are surprising. Surprisingly, they're just because of an antigenic challenge. And so, if you're able to predict very well these local sequence clusters, then you can really compare what it's there as expected and what is not. And you can, by doing that, you can extract those really weird TCRs that are much more present than they should be, and associated with the antigenic challenge, for example, in response to vaccination. And so all that I've been showing you until now is some ways that you can use these generative models to analyze your repertoires, and you can do it yourself. Yeah, there is a package online that is called SONNia. You can just pip install it. There is some tutorials in order to start using it, and I'm very open to get any feedback if there is problems or not in the installation or in the usability of the package. But in conclusion, what I wanted to show you is that there is really power in combining knowledge-based biophysical models and the expressivity of neural networks in order to characterize these repertoires. I show you that you can use that for doing many different things, and among all those, there are the possibility of looking at the maturation and differentiation of T cells, and a thing that we really want to align is that what is the difference here is that on the contrary of large language models that are very data hungry in a way, these kind of models of generation and selection, you can really look at them in a personalized way by looking at each individual and try to understand what is the difference in their immune response. And this is really what I find extremely exciting and want to look more forward and working on. And on that, I'm closing up, and thank you very much.
Thanks so much, Giulio, for this very exciting talk. I'm going to start with one general question. What is there now to be done? So what are the next steps? So what are the unknowns? What do we need to do now? And now that we have a good probabilistic understanding of VDJ recombination, where are the holes in our understanding on that front?
So we have some limit in the sense that the model itself, for example, doesn't include double-D gene insertions, and so there is some complexity that can be added on that side. So for sure, there is always space for development on the side of the model development. But for me, the most exciting part is really the fact that now this model really allow you to have a language to discuss about this repertoire, allows you to score receptors, to try to understand if those receptors are expected or not there. And so to try to interrogate the repertoire, try to deconvolve what is there just by naive reasons, or if it's something more going on there. But so it's really now the moment of trying to apply this methodology to try to better decode these adaptive immune repertoires.
Mm. And in the B cell space, there is a lot of excitement about alleles and polymorphisms, but they have been detected also or identified in the T cell space, but this has not been taken into account yet in your model, right?
No, exactly. So
Yeah.
that would be very interesting to include and to have a better resolution of the allele and the genetic background of an individual, and really whether that increase because so far the variability that we computed was really just looking at the V gene families with no really information about the alleles. And so as we include that, is this gonna create more variable repertoires, more diverse across the population. Um, and it's an interesting direction to go. Absolutely.
Yeah. And if we see that there's more variability across individuals, is it then makes does it make it more difficult to pinpoint surprising or non-surprising TCRs across individuals for antigen-specific detection or outlier detection or something like that? So does that increase the complexity
It
yeah,
does, because it makes it really difficult to like generalize from one person to the other.
yeah, right? Yeah,
And
yeah,
so like for example, even learning a big large language model or multiple receptors on a big repertoire allows you to extract a lot of information. But then if you want to really resolve this individualized part of the response, then you don't know if you can generalize in that case.
yeah, yeah,
Um,
yeah. So much more to be done. So we have two questions, Gu and then Duvi also. Yeah. So
All
Gu
right.
first. Oh.
Sure. Thanks.
Hello.
Hello. Very nice talk, Julian. Really.
Thanks.
So actually following up on what Victor just mentioned at the lyric level, I think one thing that we observed recently is like a very dramatic difference between the D2 genotype, the beta chains, which is kind of, I don't know how would SONIA look at it if it observes it by itself, but we see very dramatic differences between like basically there are two alleles in the populations of D2. So, ones that are homozygous for O1 or O2 have very different J usage, etc. So I wonder if SONIA looks at it and, in a follow-up step, I think it would be quite straightforward to add it to the model. But that's not the question that I wanted to ask.
Yeah. Fair enough.
The question that I wanted to ask is that, I mean, you showed like these difference in diversity between the different cell types in your recent publication from the models, between CD4, CD8, double positive, etc. So this is the kind of what results from the model, but did you look also the difference of the model? What is different between these cell populations? Is it the VGene usage? Is it the trimming? And did you look at the kind of more detailed?
Yeah, so I've been doing that before for CD4, CD8 still in the original paper, the SONNia paper. And there, if you're really looking at the amino acid composition, you really see a trend in polarity, and that more polars or less polar amino acids get increased selection or decreased selection with respect to CD4 and CD8. And so that this kind of opening up the box, you can do it very much with linear models, because you can really look at one of each features at a time. It becomes a little bit more complicated if you're looking at the nonlinear model. So there is on the plus side, you're able to capture more of the repertoire. On the other side, interpretability goes back. But just very quickly to answer what you were saying before, so still, even at this time, we are adding multiple alleles inside the Igor models, and so he's able to look at the relative frequencies of those. But we haven't really systematically tried to understand whether really this genetic background changes the inferred parameters. Like if I have a specific allele or another, like how is the frequency of the specific alleles in the inferred from the model is gonna be. So that one needs to still be done.
Yeah. So actually, yeah, I agree, but I think the D gene should be treated differently. I don't think that D gene is explicitly into the Igor model. I'm not sure.
It is. So we have the D gene sequence, and then we can have multiple alleles inside of the D gene sequence.
Okay. All right. So yes,
And
I'm looking forward for the results of this improved model.
Yeah.
Very nice.
Okay. Duvi?
Hey, Giulio, great talk. Pleasure as usual.
Thanks.
I had actually a couple quick questions. The first question was, so in the picture that you showed, like the figure with the scatter plot showing like the PGen versus like the incident rate of public TCRs, it looked like you had a pretty good like linear fit, like the y equals x plot.
Mhm.
What was the motivation to include the VAE component? Because it looked like you had done like a pretty good job without it.
Yeah, no, so the VAE was let me come here. The VAE was an independent realization of the model from another group. So what they did is really just looking at the receptor repertoires and training a VAE and try to see whether you can compute pigeons using a VAE. And
Gotcha.
they were doing pretty good. Just that, of course, is a little bit more data hungry, being a nonlinear generative model, and it really doesn't. And so that's why we saw that by increasing this complexity of the selection models, we're able to squeeze even more information out of these repertoires.
Okay, makes sense. Two more questions. One was, so p-post is like the post-selection probability.
Mhm.
Does it take into account the HLA allele of the actual individual? Because positive selection, negative selection, those are like very HLA dependent. Is
Yeah.
that somehow factored into p-post?
No, but in the best case scenario, you would infer a different model for each individual. So whatever p-post model you're going to infer is gonna be a data-driven model that learns the effective contribution of these HLA alleles. What would be really interesting that I started working on, but I couldn't find much on that side, is really try to crack this, doing some kind of statistical association between changes in p-post and the presence of specific alleles in an individual. And so that at least when I tried with the Emerson data, didn't work really well, or at least I wasn't able to find anything significant. Will be interesting now that are coming up a little bit bigger data sets with HLA information to retry that and see if you're really able to see differences between p-posts based on the HLA background of an individual. Yeah.
And then final question. Did you notice anything like in the data? I know it's not in like the paper like you talked about it, but did you notice anything like that's a correlation between how like polyspecific a TCR is versus its PGen? Like, is there some like evolutionary pressure to select for high PGen TCRs that are just broadly protective?
Good interest. So I think in the SONIA paper from Zachary Setna has been looking at just PGen distribution of antigen-specific receptors, and those somehow they weren't surprising. They were following more or less what we would expect just by random sampling. I personally haven't looked much about specificity, so I can't really say on that side. But there are more people working on that in the lab of Alex and Thierry. Yeah, everything that I can say on that side.
Thank you so much.
Thanks.
Okay, then thanks, Giulio, once more, and thank you to the audience also for the very nice questions. We will come back with a new seminar October 24. Both talks will soon be online on our YouTube channel also. Thanks again, everyone, and have a good
Thanks.
evening in Europe. Bye-bye.
Thank you.